Hello there, and welcome to this screencast that is going to review the processes of DNA replication, transcription, and translation. I'm going to use the puzzle pieces that we used in class so that you will be able to kind of have a way to remember those processes, and hopefully this will help you to be successful when we redo that test on Tuesday of next week. I apologize. Normally when I screencast I have a webcam so you can see my shining face, but I don't have a webcam on this computer, so you're going to have to just listen to my voice. So here we go. Alright, we're going to start out with the DNA sequence GCA. I chose to go with a really short one just for the sake of simplicity, but you do want to remember that all of the things we're talking about when they're actually happening are happening with sequences that are much longer than just 3 base pairs. Okay, so start out, here's one strand of DNA, and again our sequence is GCA, but DNA never exists as a single strand. It's always, always, always a double-stranded molecule. So here's our double-stranded DNA. This is what exists in the nucleus of your cells, and this is what is used as the template for both DNA replication and for transcription. I'm not going to talk a lot about replication because it seems like most of you kind of understand that process, but with replication you would unzip here, these would separate, and new strands of DNA would be formed, and those strands then would end up making 2 completely new but identical strands of DNA. I do, however, want to talk about transcription. The first step in transcription is the same as the first step in translation, and that is that the DNA has to unzip or pull apart. So I show that here. Here's our original GCA, CGT that has unzipped or pulled apart. The next step is transcription. Transcription is the process of making an mRNA strand from a DNA template. So here's the mRNA strand that has been built. It is complementary to our DNA. And here's the other strand of DNA. It's still here. It doesn't go anywhere. So we have our strand of DNA, we have our mRNA, which is being built according to this DNA template. Then the DNA re-zips or goes back together. So now we're back here to our double-stranded DNA molecule that we started with, and we have this single-stranded mRNA. Still, at this point, we are in the nucleus. So everything that you see here is happening in the nucleus. After this, the mRNA is able to leave the nucleus through the nuclear pores, and it goes out into the cytoplasm. And hopefully you can remember where it goes once it gets to the cytoplasm. It is the ribosome. So mRNA moves to the ribosome here. And I just drew a little picture to help you remember. A ribosome has 2 subunits, a top and a bottom, and our mRNA is in there. So out in the cytoplasm here, you can see our other molecules that are going to become important in the next step of this process, which is translation. We have tRNA here, here, and here, and then we have some amino acids here, here, and here. So what is going to happen is this is your mRNA codon, and it's in the ribosome. Okay? Out in the cytoplasm, we have this stuff floating around, and these amino acids find the tRNA that they match up with, and they bind to it. Okay? So the amino acids attach to the tRNA. Then the tRNA binds to the mRNA. So we have our mRNA strand here that was made from the DNA template. This is a codon. Remember, a codon in mRNA is a 3-base sequence of nucleotides. Okay, this is our anticodon, which is 3 bases on the tRNA. Now, one of the differences here is that tRNA is always going to be only 3 bases long. This is how big tRNA is all the time. It never has more than 3 bases. It has a 3-base anticodon always. mRNA, however, remember we talked about this as being a very long molecule. This would keep going for 300, 3,000, maybe 30,000 bases, and still it would be kind of read in groups of 3. So each 3-letter group is read as a codon. Okay, so mRNA codon, tRNA anticodon, they're complementary to each other, and that brings the correct amino acid to our sequence. And this is translation. The building of protein in the ribosome from the mRNA is translation. And once translation is complete, the tRNA is going to be removed from the mRNA, and it's going to go back into the cytoplasm, but it's going to leave the amino acid as part of the growing protein chain. So in this example, this is our first amino acid. It's arginine, so it doesn't really have anything to bind to. But this, remember, would be much longer if this were an actual mRNA molecule. So another tRNA would come in here with another amino acid, and that amino acid would bind right here. And we would just keep adding amino acids one at a time until we got to whatever our protein was supposed to be, 100, 1,000 amino acids, whatever it is. Okay, so I hope this has been helpful to you. I hope that it will help you to better explain the process on our test next week. If you have any questions, be sure to let me know. Thanks.